Hi there, my name is Mary Beth Stafford, and today I wanted to share my research on the survival rates after cryopreservation in the Hawaiian species Melicope mucronulata, more specifically, the impact of genetic variation in tissue age. I'm a University of Cincinnati graduate and have worked at the Center for Conservation and Research of Endangered Wildlife, or CREW for short, for 3.5 years. We're located at the Cincinnati Zoo and Botanical Garden. And this past year, I received an award from BGCI that allowed me to fund my own project and design my own experiment after our IMLS grant ended. So I drew the illustrations that you're seeing in this presentation to better explain the cryopreservation process. And part of the IMLS grant that I mentioned worked on cryopreservation of endangered Hawaiian species. And the species that I worked on is named Melicope mucronulata, and it's endemic to Hawaii on the island of Molokai. Presently, there are 2 plants left in the wild, so at CREW, we worked with the Hawaiian Arboretum in Hawaii to take their tissue culture collection, cryopreserve, and bank the tissue here as a genetic backup. And here's a photo of Melicope in the wild. It's a woody shrub that grows in a fairly high altitude, so temperatures can get pretty cool, and it's not so tropical as you might think. The problem with Melicope is that it doesn't create very many viable seeds. And so in terms of plant terms, we call this an exceptional plant because traditional seed making methods don't work. So instead, we work to preserve the meristematic tissue within the nodes of the plant because this tissue is capable of growing a whole new plant, sort of like a stem cell in a human. So if you get lucky, sometimes you can use the same protocol that you used on one species with a different one, especially if they're in the same family. This genetic variation within individuals, it's what's called a genotype. So the challenge with cryopreserving Melicope is that survival rates differ by individual and not just by species. So this is an issue because we only have a few genotypes with high survival rates that we can bank plenty of, and then conversely, several genotypes we struggle to preserve. This isn't good because if we bank what we can, when we take the plant out of cryopreservation to reinstate the species in the wild, should it go extinct, we've artificially selected for some genotypes and not others, which could create a whole host of problems. So before we talk about my BGCI experiment, I wanted to briefly show you the cryopreservation steps, and that way you can see why protocol development is so crucial and what variables affect survival. So once we receive the tissue from the Hawaiian Arboretum, we propagate the plants to get a bunch of tissue. You can see that photo on the left. We start with one test tube and we make a bunch. Next, we isolate shoot tips from the plant and we put them onto pre-culture medium, and this dehydrates them before cryo. Here's an actual size of our Melicope shoot tips. You can see they're very, very small. Next, we put the tips into a few different solutions. So the whole goal of this process is to remove as much water as possible because if ice forms, it'll puncture that cell wall and then it'll kill the plant. So the vitrification solution that you see down at the bottom of the screen, that kind of acts like a plant antifreeze. So we put those 10 shoot tips in a little aluminum foil boat. We submerge it into liquid nitrogen and then put it in a cryo vial where it can be stored for years. If we want to take the tips out, we rehydrate them and they recover on nutrient medium. So in theory, the plants can be sent back out to Hawaii and restore the native population like I mentioned previously. The reinstatement step requires a whole host of factors since plants coming out of the test tube go from 100% humidity and all the nutrients they need fed to them via gel to low humidity and soil. So this process in and of itself can take weeks to months. This leads me to my experiments. So last January, I preserved our collection using 2 different protocols for cryopreservation. Our standard CREW protocol, labeled CREW, and then a USDA protocol for citrus plants since Melicope is in the citrus family. And the green boxes show survival on CREW pre-culture medium on the left and then recovery medium on the right. And the orange boxes show survival from the USDA pre-culture and then subsequent recovery medium. And so you can see that survival varied by pre-culture and recovery medium, and then a combination of the USDA pre-culture and the CREW recovery medium being our ideal catch-all for the future cryo experiments. So this paved the way for my BGCI experiment because I now had a protocol in place that would hopefully improve survival odds as a whole moving forward. So the orange boxes signify where the experiment in January modified my protocol. You can see the experiment started in the beginning and the end of the protocol, but where my BGCI project comes into play is in that box in blue. So instead of changing the media, I was interested to see the impact of tissue age at the time of isolation and then compare that to survival rates. So this is because Melicope has been shown to have differential survival not only by genotype, but also at the time of isolation within that genotype. So in a preliminary experiment, we used two genotypes that were cryopreserved. We used a 15-day-old explant and an 85-day-old explant. And for both genotypes, survival of the younger tissue was higher. So this made me interested, and I wanted to do a larger scale experiment. So to investigate further, I subcultured 4 different genotypes of Melicope on the same day, and then I isolated them at 2, 4, 6, 8, and 10 weeks. So that photo on the right is a 4-week-old plant on the day of cryopreservation. So because of the nature of endangered plants and collecting tissue, my data this time around was the data set itself was very small. So I'm not surprised I didn't see a statistical difference, nor did I really need one. I'm just really happy with the trend that we saw. So you can see on the graphs, there's 2 different genotypes, which survived cryo at different rates. And each kind of has the beginnings of a rudimentary survival curve. So you can see genotype A is in light green, genotype B is in dark green, and you can see it starts to increase in survival, and then there's a sharp drop off. So we previously thought that the 4-week-old tips would survive the best, but now it seems like the optimum time to isolate might be around 6 weeks. So survival for the genotypes in and of themselves is very low, but for these 2 genotypes, we do see that upward trend in survival, and that corresponds to tissue age at the time we isolated them. So ultimately, this highlights the importance of staggering the dates of cryo in order to ensure that all the tips that we're preserving are preserved when they will survive the best. So here's one of the genotypes growing on recovery media after cryopreservation. You can see at that photo on the left, you've got the very nice, like normal looking leaf and then a whole bunch of callus. So there's a ton of reasons which this could happen, which we're really not sure why that happens, but sometimes plants in tissue culture generate a lot of callus. Anyway, so Melicope was initially banked in 2019, so samples have actually been in cryostorage for 2 years. And this made me interested in sample longevity because we only leave the plants in liquid nitrogen for an hour, which usually it doesn't impact survival too much that we've seen, but we haven't had this amount of time in order to analyze them before. So I was very excited to take some genotypes out and figure out the impact of survival in that way. So I took almost, I think, 6 genotypes out roughly, and I recorded their initial survival data in 2019, that's the light green, and then I compared that to survival when they were removed in 2021. And so you can see those little boxes kind of show the genotype before, the genotype after, and so for most genotypes you can see a decline in viability, but an increase in this genotype right here. And this again highlights the varying survival rates within the species and the many factors that contribute to it. So cryopreservation of shoot tips began after 1970, so we don't really know much about long-term viability. So in terms of Melicope cryopreservation, there are duplicate samples that are still in storage, and I've put many in there myself, and we can evaluate those in the future and hopefully provide some data points regarding rate of decline. So I think of this experiment as like a second data point in terms of a long-term project. And here's a sample banked in March of 2019 that I removed from cryo in late 2021, and it survived to tell the tale. In terms of next steps, there are so many endangered plants which could benefit from a genetic backup so that we don't lose them forever. And I think the experimental design of the 3 projects that you saw today can act as a template for cryo experiments going forward, so that at the very least we're able to mitigate those potential variables and we can navigate them more efficiently. And because of these variables, they require a lot of time and money. So I wanted to thank Abby Meyer for thinking of me and everyone involved in the award process. Crew's grant wrapped up in November, so without your contribution, I wouldn't have been able to do the research that you saw today. Thank you very much. Have a good day.